Hello, and thank you for joining us. Today's webinar is Back to Basics, an overview of the TOC analyzer role in pharmaceuticals. Our presenter today is Tyler Trent, Product Marketing Manager of the Elemental business. If you have any questions, please feel free to submit them and we will get back to you offline. This webinar is being recorded and will be available within a few days. Okay, now I'm going to turn it over to our presenter, Tyler Trent.
Thank you for that introduction, Shelly. As Shelly says, my name is Tyler Trent. I am the Product Marketing Manager for the Elemental business here at Teledyne Tekmar, Teledyne Lehman Labs. Today, we're going to be talking about a topic of back to the basics, an overview of the TOC analyzer role in pharmaceuticals. First, just some housekeeping here. Let's go over a little bit of an outline. First, we're going to talk about who is Teledyne. Sometimes people might wonder what Teledyne does. So I'll explain a little bit about that today. And then what is Teledyne's role into the pharmaceutical market? So where do we play? What impact do we have? And where are we have instrumentation at through the drug development process? Then once we're completed with that topic, we'll be looking at the TOC analyzer, specifically the Fusion TOC analyzer from Teledyne Tekmar, which is the UV persulfate system. So we're going to do an overview of the system itself. Then we're going to look at a couple methods, cleaning validation or CIP, and also USP 643. And then we're going to dive into maybe a real world application and kind of talk about the differences of the techniques we're talking about here today. So Teledyne Technologies, our slogan is everywhere you look. It's actually a good slogan for us because we are actually everywhere you look. We have many different divisions. We have an instrumentation division, digital imaging division, aerospace, defense, electronics, and engineered systems. So basically on the sea floor, ocean surface, across land, under the ground, in the air, and in space, Teledyne is everywhere you look. As you all might know, the Mars Perseverance rover was recently just landed, and here's some of the Teledyne products that are actually on the Mars surface right now, collecting data and sending it back to Earth. So as you can see, we do a whole host of different things. But what we're going to be talking about today is Teledyne Pharma. So Teledyne is a leading instrument manufacturer with products spanning the entire spectrum of the pharmaceutical process. So we look at drug research, development, manufacturing, QC, and beyond. So what you kind of see over here on the right-hand side of the screen, if you follow my pointer, is a whole host of instrumentation or systems that we use throughout that process of research, development, QC, manufacturing. So what we might see here is a dissolution system, some automation systems, flash chromatography, also the TOC analyzer, which we'll be talking about more today. So you might ask what type of companies are involved from Teledyne into this pharmaceutical business segment. Well, first is Teledyne Tekmar, which is going to be the TOC analyzer. We got Teledyne ISCO, Teledyne Hansen Research, and Teledyne CTAC. We'll take a little deeper dive into those companies right now. So what we'll see here for the Tekmar side is volatile organic compounds, VOCs, and also the total organic compounds, total organic carbon compounds, TOC analyzers. That is what is produced here at Teledyne Tekmar. Then as we look at a couple of different other companies, as I mentioned in the previous slide, Teledyne ISCO makes small molecule purification, precision pumping, and then we go over to Teledyne CTAC, where they actually do some of the development or R&D, where they're looking at low volumes, micro volumes, sample introduction types of systems. Then our main company that does a lot into the pharmaceutical is Teledyne Hansen. They have everything from dissolution testing, diffusion testing, tablet physical testing, sample preparation, and other types of instrumentation. So as you can see, all these four companies right here make up a larger group called Teledyne Pharma. It's a big marketing segment so that we can address the customer's concerns from development to manufacturing to QC and beyond. So we have every stage of the process covered with this group of companies here at Teledyne. So I want to talk about the Teledyne Tekmar TOC history. We've had a long-standing history of TOCs here at Tekmar, Teledyne Tekmar. It actually goes back to the 1960s to a company called Dorman. As time went on, Dorman was sold and became Tekmar Dorman, then it became Teledyne Tekmar. The Dorman name was dropped. But since then, Tekmar has evolved from Dorman. We have had 5 generations of TOC combustion analyzers. So that's for your high-level, dirty matrix types of samples. And then we have 4 generations of the TOC UV persulfate analyzers that is basically looking at drinking water, pharmaceutical water, water for injection, drug manufacturing process, and those types of applications. And each generation, as we change over time, has demonstrated vast technology improvements over its predecessor. So as we can see down here on the bottom of the screen, we start off with a very simple TOC analyzer, and then we move through the time with our current portfolio being the Torch, which is a combustion analyzer, the Fusion, which we'll be talking about today, which is the UV persulfate analyzer, and the Lodix, which is the low-cost, simplified TOC combustion analyzer that we have in our product portfolio. So as we look at the Fusion TOC analyzer, as I said, this is the 4th generation UV persulfate system that has come from Dorman, Tekmar, now Teledyne Tekmar. It combines the analyzer and autosampler into one. So no need to really worry about having a separate autosampler or getting a separate autosampler quoted. It's actually all in one, all-inclusive type of system. So a little bit about the Fusion. So there's a couple of different cool features that we have on this system. We have auto calibration. So basically, if we look at the system, within our sample rack, we have 475 ml vial positions or 125 ml vial positions. Those are actually where we have our standards or our check standards. The system will automatically dilute those from a single stock solution, eliminating the need for manual preparation, which also reduces time and also reduces the likelihood of human error. That's all possible due to the syringe that we have here that we can do dilutions on the system. One other great feature is IntelliDilution. So if your sample that you're running is currently out of range or over range is your calibration curve, if this feature is checked, it will actually see that the sample is overranged. It will come back, dilute that sample. Sorry, first it will rinse to clean out the system. Then it'll come back and dilute that sample. Then it'll try to bring that sample within your calibration range. It will try 3 times. This eliminates the need to manually dilute the sample and rerun. So that's a good little feature here. Since this system is a pressurized system, we have a mass flow controller. A lot of the other TOCs out on the market are set fixed flows with needle valves and different things like that. But our system is controlled. The Fusion is controlled by the mass flow controller. Another Teledyne company makes this product for us. It allows for higher flows between samples to reduce for cleanup times and optimizing sparge flows for each sample. It also helps to reduce the gas consumption because when we're done with it, the mass flow controller will turn off and no more gas is flowing through the system. So you're not wasting any type of gas or running your tank dry overnight. The system actually will shut it off completely. Another good feature about the Fusion is 21 CFR Part 11. We provide you tools for compliance. We don't do the compliance. That is based upon the laboratory itself, but we provide you the tools within our software. This comes standard. This comes standard with every Tekmar software if you buy the Fusion or the Torch. It comes standard in that, and it doesn't require any additional charges for the disk. Then another thing is we have an IQ, OQ, PQ package that is written and revolved around the pharmaceutical process for this 21 CFR to help you with your processes or for your audit capabilities. The system also comes pre-programmed with methods. So this basically takes the guesswork out of method development, as it's already pre-programmed into the system. So we have methodologies for drinking water, wastewater, USP pharmaceutical, Japanese pharmaceutical. We have different methods programmed for you with what we determined during development was the optimal parameters. But you as a customer will also be able to be afforded to develop or change the methods parameters that you want to change to. And then, as I said, automatic shutdown. It saves an incredible cost of money due to less gas consumption, and that is due to the Teledyne Hastings mass flow controller that is controlling our gas flow within the Fusion system. So this is kind of a high-level view slide that we're going to be looking at today. So the key testing points for TOC in a pharmaceutical manufacturing process are what? We're going to be looking at incoming / source water. We're going to be looking at the reactors and waters used during the cleaning process, and the production of ultra-pure water for injection. Then we're also going to be, as we go in deeper into the methods, which was one of our topics today, we're going to be looking at clean in place, CIP, or cleaning validations with swabs. A method that we're not going to be touching on, but we will go over a little bit, be water for injection, WFI, or ultra pure water. And then we're going to specifically talk about USP 643. Those are the key applications that this system is made for, along with drinking water for environmental purposes. So, as we look around our pharmaceutical manufacturing laboratories and different things like that, we'll see many different types of systems. A lot of the manufacturers actually have many HPLCs out there for routine screening. So, is TOC better than HPLC for routine screening? We'll find out in this presentation later down the road, but when we start looking at the high-level overview of it, it's a low system cost. TOC has a low system cost compared to HPLC. It's a quick analysis, 5 to 10 times faster than other techniques. No sample prep or minimal sample prep is required. Little to no method development, and low cost of operation, reagents, service items, and consumables. The TOC is a pretty simple machine compared to an HPLC or high-performance liquid chromatography, where you have pumps, tubings, columns, buffering solutions, and degassers, and different things like that. So it's a much simplified choice. And then we'll talk about a little bit about the benefits of the Fusion TOC system. TOC can be detected in any carbon-based substance. Makes it great for a general test. Great sensitivity, sub-ppb levels, easy, quick sample pathway cleaning, accurate analytical results, few interferences. There's a lot of well-documented published methods out there for this system. And we can provide a large analytical range. So for the Fusion TOC can go all the way up to 4,000 ppm of carbon. It's all dilution dependent. So when we start talking about these applications here, we're going to be looking at clean in place, cleaning validations with swabs, and then also USP 643. So cleaning validation with swabs, what is that technique or what is that method that we're going to be looking at? So a little bit of background. It's a multiple-step process to ensure cleanliness within the pharmaceutical manufacturing process. So the FDA has published guidelines on cleaning validations dating back to 1963 under certain regulations. So firms or drug manufacturers must make sure undesirable compounds are not being transferred from one batch to another batch using the same manufacturing equipment, because we know a lot of manufacturers make multiple different drugs in the same equipment. So they have to ensure the cleanliness between batches. So pharmaceutical cleaning validation, what does it involve? It involves a two-step process. The two-step process is analysis of TOC in the rinse water, and then also TOC analysis of the swab samples. Some people might ask, how do you swab, or how do you do certain things revolving around the swab? So I'm going to take you through a step-by-step process. So for our purposes, we use in our laboratory a section of stainless steel or plastic coupons, which are these little guys right here. They're a known surface area. And basically what we did, or what we are doing, we soiled it with a chemical of interest. So we're looking at something within the process to observe if our cleaning process is basically good enough to allow us to continue on with the manufacturing process. So it's soiled with a chemical of interest and swab clean to observe the efficiency of cleaning validation using the swabs. So how do we swab? So swab sampling, part 1. So for us in the laboratory, as I said, we use these stainless steel coupons, and then we basically apply a volume of a stock solution to the coupon itself. So we know exactly the concentration. We allow the coupon to dry, simulating the cleaning conditions as much as possible. Then now we're going to do the swabbing of the coupon using the proper technique, or a proper technique that's been out there. So first, what we want to do is we take our swab and we swipe down. Then we're going to flip our swab and swipe up. We're going to flip our swab again and swipe down. Then we're going to do zigzag from corner to corner, flip again, and do zigzag from corner to corner to ensure that we're getting all of the surface area onto the swab. Then from there, we have a 40 millivella vial. We cut the swab tip off, place it in the vial with ultra pure water, shake a vortex. So that's going to release the carbon from that swab into the liquid sample, or into the liquid diluent itself. Then we're going to put that vial into the Fusion TOC analyzer and run the system and analyze that vial. But there's other steps that we have to take into consideration so we can ensure accurate results. We must determine some background. So what type of background is happening? Do we get background from the swab, background from the DI water or the ultra pure water being used, different things like that. So we fill a vial with low TOC grade water, close the vial with a cap and clean septum. In a separate vial with partially filled with low TOC grade water, we soak the swab. We express any extra water out on the side of the vial. We clean a coupon. We swab a coupon using the same process that we talked about above, but it's not a soiled coupon. It's a clean coupon with little to no residue on it. Same process as I said before. We swipe down. We flip the swab, go up, flip the swab and go back down. We do corner to corner in a zigzagging motion. We break off the swab tip inside the vial. We put the cap and septum back on the vial, shake a vortex, and then we analyze that for our background on the TOC system, which will be the UV persulfate fusion that we have here on the screen. So a little bit of conclusion about that process and how that really works is it's a multi-step process that has to be done in different stages. So what you saw above was basically the analysis of the swab. But the more simpler side of that, or the easier side of the two-step process, will be analysis of the rinse water. So as that rinse water is exiting the vat of the tank, you're collecting a water sample and you're analyzing that for TOC concentrations. And that could render different undesirables inside your tank that might need to be cleaned out further and further until you are at your approved levels to move forward to your next batch. So the other method that kind of goes hand in hand with this is USP 643. So what is USP 643 and where is it used at in the process? So it's used to test the system's ability to oxidize different organic compounds. And what you might ask? Well, bulk water. Bulk water. What is bulk water used for? It's used for water for injection or maybe purified water that is used for the process. It's also used in sterile water. So once again, sterile water for injection, sterile purified water, sterile water for irrigation, or sterile water for inhalation, such as inhalers and things like that. So it requires certain levels to be met to be used within our pharmaceutical products. This also could be done for cosmetics or personal care items, but it's not required. So what are the requirements that we look for in USP 643? Well, we have a limited detection, LOD, at 0.5 ppm in bulk water and 0.1 ppm in sterile water. Our reagent water is 0.1 ppm. Our sterile water is 0.5 ppm of carbon. Standard solutions, so sucrose is a big proponent of the USP 643 requirements, and also 1,4-benzoquinone is also a big requirement for that testing. So bulk water, half a ppm or 0.5 ppm of carbon. But if we look at sterile water, it's increased up to 8 ppm of carbon. Then we also have to look at a certain response efficiency. So the response efficiency is looking at the oxidation capabilities of said system or the UV persulfate fusion system from Teledyne Tekmar. So the standard requirements are anywhere between 85% to 115% response efficiency. So you might ask, what is response efficiency? Sorry, let's talk about the difficult to oxidize first. So we know, looking back on the screen, for these requirements to be met, we have to analyze a certain type of solutions. So a sucrose solution, a benzoquinone solution, and a KHP solution. So as we look at the screen here, moving from left to right, we have difficulty of oxidizing compounds. So sucrose, very easy to oxidize. KHP, that's what we make all of our TOC standards from. And then it's a little bit more difficult to oxidize, which is 1,4-benzoquinone. So back to what I was talking about response efficiencies. So how do we get that, or what is this really telling us for our measurement here? Well, we have to measure a couple different things, and this is the part of the USP 643. So we have to look at the instrument response, RSS, which is instrument response to system suitability. Okay, so that's what we are looking at here for 1,4-benzoquinone. RW good linearity, 0.9995. And that's what we receive from our raw values and our expected values and our slope of our line. All documented on this page. And then basically when we start looking at our system suitability, it's actually going to have a detailed report for you about your system suitability. And this is just a clip or an excerpt from that report. So what it's going to do is going to look at your reagent water. So your reagent water is no higher than 100 ppm. All right, my reagent water for this study was at 0.14 ppm. That passes. That's perfect. Then I look at my system suitability stuff. My standard sucrose solution, half a ppm, came back at 0.57. My 1,4-benzoyl came back at 0.56. Well, what does that mean through my calculations to calculate my response efficiency? Well, my response efficiency for this system, for that run on that day, was 97.75%. That means right then and there, our system is good to go to continue on with the validation or running of the samples that day. So when you start looking at the system suitability, you're going to have to run it once a day, twice a day, depending on your SOP. And this software actually has these built-in tools or built-in wizards that allow you to populate it automatically within your schedule when you select the drop-down of system suitability. And you have complete control over your pass-fail criteria as a laboratory manager or a quality control person. You can go in and do that, and then you can set permissions within the software so a user or the analyst can't go in and change anything. All they can do is load the system and press run to go, and that's already been populated for them. They don't have to do anything. You don't have to have any worries about changes to the method, changes to the system suitability. It's all locked in, especially with permissions given within the 21 CFR software. That's another great thing about the Fusion system is that when it comes to pharmaceuticals, we can set up different accounts. So we can have an administrator, we can have a laboratory manager account where they can go in and modify methods, modify pass-fail criteria. That's all they can do. Then you can have a laboratory user or an analyst method or analyst setting where they can just create a schedule and press run and go. So we offer different levels of protection from the administrator all the way down to the system user. So when we start talking about real-world examples here, a lot of times we get compared to HPLC results. Or what do we look for? Because a lot of times a laboratory or pharmaceutical laboratories have HPLCs that are relatively available, okay? So back to this main slide here, is TOC a better choice than HPLC for routine screening? What I like to do is take you through some questions here and also show you a real-world example to see what your thoughts are. So here's our title slide for this section. Advantages of using TOC and cleaning validation from an analytical perspective. So as manufacturers of drugs and biologics, we must consider active ingredients and compounds in the products to limit establishments. HPLC, high performance liquid chromatography, with certain types of detectors, if it's a UV detector, photodiode detector, or a mass spec on it, is often used for testing purposes that give the best system response. Well, why is that not okay sometimes? So when we start looking at the cleaning process, are the only ingredients, active ingredients, a possible source of contamination? Are they coming from the cleaning agents, the active ingredients, the byproducts, the excipients? They all contain TOC, so we can get a good reading. But is that just our only source of contamination? No, that is not our only source of possible contamination. Possible contamination can also come from outside sources, such as from human intervention or other objects. So if it's a possible source of contamination, would it be more effective to have an instrument that can test for all sources of contamination and not just highly specific compounds? Because you never know if something might happen or an outside source could do something to that batch, and you got to figure out where it is, and that's going to be a long analytical process. So yeah, we want something that can tell us, is this contaminated or is it not, or is it clean? So when we start looking at different types of methodologies, so we have TOC, high performance liquid chromatography, then TLC, conductivity, and spectrophotometric techniques. Well, are they specific or not? TOC is a non-specific method because we're going to analyze carbon. We don't care if it's certain types of compounds. We just know there's carbon there, and carbon shows activeness of biological. Carbon is the basic building block of a lot of things. So we want to make sure that when we do pharmaceuticals, especially for water for injection, where it has to be less than 50 ppb of TOC, we are analyzing it to the best abilities that we can. So TOC here, not a specific method. It has a broad analysis with low-level detection. And disadvantages, it won't tell you what compound it is. It will just tell you that it's carbon. HPLC, which we're comparing this to right now, is a very specific method because you're going to set up retention times. You're going to be setting up different types of standards that tell you where that compound's coming out. So it's highly specific. It's going to give you a lot of those need to know details. But there's some disadvantages. It's a long run time, expensive, requires a lot of analytical knowledge, and sometimes it's too specific. Sometimes you need a broad-based analysis that gives you the superior detection limits, and that's not HPLC because it's so specific. TOC is going to detect any carbon-based substance. So when we start looking at this, which part of the capability, which part of the methods or different things like that can be used? Drugs, cleaning agents, drug residues, cleaning agents. So what we see here is TOC is a yes across the board. It can be used for all three of these. HPLC can only be used for 2, TLC can only be used for 2, and spectrophotometric, depending on what you're really looking for, yes or no, yes or no. Well, that's because of different types of interferences and different things like that. So what looks to be good, or what looks to be the most desirable for what we're doing, is going to be TOC. So TOC has grown in acceptance for cleaning validation analysis. It's typically reserved, an application is typically reserved for a single product assay like HPLC, but the methods can be developed, validated, and used efficiently in pharmaceutical cleaning validations. And over time, there's been a lot of great publications. This is a publication right here. TOC has a low-level detection, rapid analysis time, is low cost compared to other methods and can detect all carbons-based residues. This is a great application. This is a great paper. So if you can, go out, read it, and it allows you to understand a little bit more about the use of TOC in the pharmaceutical process. So when we start looking at TOC and cleaning validation, well, TOC has a low system cost compared to HPLC, anywhere between 5 to 10 times lower. Very quick analysis. So where a one run on an HPLC system could take up to 30 minutes, so you can get all your separation, TOC can get 1 replicate or 3 replicates done within 20 to 25 minutes. So you're going to be getting faster sample analysis and replicates compared to 1 single run on the HPLC. Minimum sample preparation is required. So for TOC, you basically collect the vial of water, place it into the system. Sometimes people might preserve it, but you don't have to if you're sampling and running it that day. But with HPLC, you gotta get the sample, You got to dilute it in your buffers. You got to make your buffering solutions. You got to do a lot of different stuff. And also method development. You've got to be able to dial in the retention times on HPLC, where TOC is basically take the sample, sparge it, remove out the inorganic carbon, place it over into the UV reactor chamber with persulfate and acid. That's going to promote the oxidation of carbon to CO2, and it's going to be sent over to a CO2 detector and measured for our concentration. It's a very low cost also. It's a very low cost of operation, as the reagents are used for the system are very inexpensive compared to HPLC columns that could be large pour, small pour, or they could range anywhere from $500 to $1,200, maybe more, depending on your molecule size. So with TOC, it's a very inexpensive cost to run and operate the SAM system. TOC, not specific. TOC detectors or any carbon-based substance, so it will see contamination from all sources. So if it's coming from the vat itself, from the water used in the cleaning process, or one of your reagents, it's going to be able to detect any carbon-based substance. So that's good news. And if there is low TOC, the possibility of contamination is remote. So we can use that as a pass-fail criteria, go or no go. If the TOC level is high, then other testing can be used to determine the source of contamination. But the end result is you still must clean again. So if your TOC level is high, you can continue to investigate where that's coming from, but you still have to clean your vats. You're going to have to clean your processes again. So that's a good thing to see. The same instrument can be used for TOC testing of pure water, water for injection. The fusion system can be used for many facets within the pharmaceutical process other than cleaning validations. So here's a real world example that we got from one of our customers back in the day. So the data is volunteered by one of our customers of a system that revealed the TOC value obtained from one cleaning process, cleaning analysis, exceeded their limits. An alarm was set off. So what we see here is they have a certain TOC result and a certain TOC limit. For the first row here, the limit is 1.1 ppm. For the next sample, it's 2 ppm. For the third sample, it's 2.0 ppm. So let's analyze our water for the process. Well, the first one is 304 ppb. Does that reach our limit? No. Any action required? Nope, we're good. Next one is 21.9 ppm of carbon. Well, that's well above our limit. So is action required? Yes. So what must we do? So they ended up using, taking the sample over to an HPLC, used as a supplemental specific technique in the event of a failure. That was performed on a backup sample, but yielded no additional information because it wasn't in their confirmation. So they had to further do discovery. They had to further understand what is this compound or what is this undesirable in my process. Well, they ended up taking a second sample of the TOC, and it yielded the same results, very similar to the first one, 21.2. 2 weeks of investigation eventually proved that the contamination source was hand sanitizer that was used by the person collecting the sample. So this right here on HPLC HPLC is a specific technique. If you don't know what you're looking for, it can't be that specific and you have an unknown. And you have to further do investigation. But when we look at TOC here, we know that we have a go, no-go, or actionable limit that we can set. 304 ppb is below our limit of 1.1 ppm. No action needed. But as you see on the second sample, action was needed, but further investigation proved it was an outside source of contamination from the personnel collecting the water. So what does that tell us? The contamination was basically eventually identified by LCMS, which is a more expensive technique than TOC. So with several modifications, extensive method development for identification, it was able to be discovered that it was the hand sanitizer that was contaminating their process. But with TOC, one simple run was able to identify that contamination and brought to their attention very, very easily. And they didn't have to, they continued to go through the process of tracking down the contamination. But if you don't need to continue to do that step, a no or a yes, a no-go or yes-go step for your actionable limits is great because TOC can work to identify those for you. So this case was a simple contamination, was not system contamination, it was external contamination. It was clear to the customer that the TOC saw the contamination that the HPLC would have never found, because it would have found it, but it would never have been able to identify it. They had to continue to do method development to identify what was needed. So this incident validates the power of TOC in the cleaning application to detect contamination from outside and internal sources within the development, within the process of manufacturing. So some disadvantages here when we start looking at the HPLC compared to the TOC. HPLC is used primarily just because it's available in the lab. There's a lot of them available. Cleaning validation sometimes tie up the HPL system just to do CIP work. Wouldn't you want that HPLC system to be doing other things? The TOC can do all that cleaning validation work for you. Many companies have budgets for new HPLCs to cope with existing workloads because the sample workload is being taken up by the clean in place or the CIP. So by incorporating a TOC with 90 position carousel, different things like that, it's going to limit that workload for the HPLC and free it up to be used for what it needs to be used for. As I said, run times are 30 minutes versus 3 minutes by TOC per replicate. If you include all the rinses and doing it by triplicates, you're anywhere from 15 to 20, 22, 23 minutes for one sample on the TOC with triplicate analysis compared to one run on the HPLC for 30 minutes. There's method development. There's possible mobile phases, columns, so costly time setup, costly consumables, different things like that. One thing about the HPLC, it's more likely to be questioned in an audit than TOC, as current auditors have very good working knowledge of the HPLC and can find conformities much easier than with TOC. So some of the advantages that we look for with TOC is, it's a system cost. Everyone is cost conscious these days. They need to be understanding their laboratory budgets, how much they're spending in consumables, how much are they spending in the capital purchases or system purchases. Well, TOC is 2 to 3 times less expensive than an HPLC. That's your base HPLC. If you start going to HPLC mass spec and different things like that, the cost savings are going to be even more. Better to originally purchase TOC for CIP or cleaning validation samples or clean in place samples than HPLC, because it's going to free up that system. It's going to allow you to get easier, faster results and be able to hit your actionable limits. Yes or no, can I move forward with this process, or do I need to go back and reclean? Faster runs mean better turn times. So when we start looking at the benefits of the Tekmar Fusion TOC analyzer, one great thing about operating in a TOC analyzer, it can detect any carbon-based substance. It's great sensitivity on the Fusion, sub-ppb levels. So we're working down at the 50 ppb for water for injection, all the way up to 100 ppb for different things, maybe up to a few ppm. So we have great sensitivity, but we also have flexibility within our analytical range. Easy and quick sample pathway cleaning. With the Fusion, all the glassware is removable. So if it needs to be cleaned, it can be easily cleaned in nitric. And the system also does system rinses and different things like that. So it's easy to set those up and clean out the pathway. accurate analytical results in great and fast time. We all want great results, but we also want them done in a timely manner. Few interferences. Some different other techniques out there might have interferences, might not be able to handle certain things within the process, or might have additional modules that are needed to remove IC and different things like that. So the Fusion TOC system has few to little interferences. It's a well-documented and well-published methods out there, and it's also a large analytical range. So we can see the Fusion can be used from the sub-ppb levels up to the ppm levels to encompass everything that we need to do for our pharmaceutical validation process, cleaning validation, USP 643, and also other processes such as butterfly injection and other bulk or sterile analysis that we have to do. So with that, I want to thank you guys for your time and allowing me to explain a little bit about the cleaning process, how to use swabs, what are we testing for within the process itself, such as the cleaning water and also the coupons or also an area of the tank. And then moving into the explanation of the USP 643 with a little bit of information about the 21 CFR capabilities of the Fusion itself. As Shelly mentioned earlier in the presentation, that if you have any questions, please go ahead and submit them and we'll be able to answer them offline to you in a timely manner. So thank you for your time today.